Hello everyone, I am Yash Parvapuri. Today I will be presenting my bioinformatics project which I did at biotech company under the guidance of Dr. Raghavendra Lakshminarayanan and Ms. Sonali Kari. The vision of biotech is to enhance human health and well-being by enabling biological research and discovery with relevant data, solution and support. The title of my project is Gene Expression Landscape Rebels Pathways Associated with Parkinson's Disease Progression in Age Match Comparison using transcriptomics RNA sequencing based approach. Currently, I am a student of MIT University, Mumbai, and I am currently doing my bachelor's in biotechnology. My field of interest is neuroscience, genetics, plant biotech, bioinformatics, and also I have interest in transcriptomic profiling and genomics. So, what exactly is Parkinson's disease? Parkinson's disease is the most common motor neurodegenerative disorder resulting from the loss of dopaminergic neuron in substantia nigra. The classical symptoms of Parkinson's disease are tremors, bradykinesia, and dystonia are expressed as motor control abnormalities. Although a substantial body of research focuses on a central nervous system which affects effects of Parkinson's disease, very little remains understood about peripheral neuromuscular defect. Recently, transcriptome profiling via skeletal muscle transcriptome wide RNA sequence among individuals with Parkinson's disease and healthy old and young control subjects are actually done in the present research which I did to compare the disease progression of Parkinson's from the omics and transcriptome point of view. So for choosing the good data set, I searched the publicly available data and come across this interesting data set, which includes human subjects, Parkinson's disease samples, 12 samples, old adult sample 12, and young adult sample 12. Both old adult and young adult were non-exercising disease-free control subjects. Transcriptome wide skeletal muscle RNA sequence are approximately 60,920 transcripts. This data set is very interesting because it not only compares the Parkinson's disease patients with normal control sample, it separately compares the Parkinson's disease patients with control sample data old age and the control sample data young age. So we can split our comparison and understand and focus specially on those genes of pathway that are responsible for Parkinson's disease progression. That's why I choose this interesting data set. The dataset which I took for analysis is a normalized dataset, not the raw data. bioinfo is a bioinformatics platform that combines statistical analysis models into a pipeline to deal with heterogeneous big data. It enables faster and easier analysis, integration, and visualization. It is algorithm-based web interface. It builds a high-throughput data processing pipeline. Here we perform different pipelines for PCM and DSEC2 enrichment analysis and GSA analysis. These are both the pipelines for both the analysis. The total number of transcriptome wide skeletal muscle RNA sequences is 60,920 transcripts. Then these samples are separated based on their age, that is, young adult and the old adult. After sorting out with details, we got 31,417 transcripts of Parkinson's disease versus old adult and 19,047 transcripts of Parkinson's disease versus young adult. And then these values are then sorted with respect to p-values, by which we got some significant number of genes. So the data is like we got 1517 significant XL gene in case of Parkinson's disease versus old adult and 387 significantly XL gene in case of Parkinson's disease versus young adult. Currently, we don't know which are upregulated and which are downregulated. In the further steps, we will know which are upregulated genes and which are downregulated genes. Now I will discuss the result of the analysis. Firstly, I will discuss principal component analysis of PCA. It's a dimensionally reduction method that is often used to reduce the dimensionality of the large data set by transforming a large set of variables into a smaller one that still contains most of the information in a large set. Here we can see that PCA has a quite high coverage. If we add PC1 and PC2, we will approximately get 63% of PC. But despite that data is well separated in PD versus OA. Colors and data points are mixed with each other. So, according to our analysis, the PCA doesn't give away or doesn't show very well set of separation between two sets of samples, that is between PD versus OA and PD versus Y. But usually we would do something that would require to change. But here with the differentially gene expression analysis, the thinking and understanding that this data might not be conducted on this data with the linear approximation or linear dimension reduction that PCA applies to that data might not be reflected in the separation between PD patient and the young adult and the old age patient. Then we perform the de facto analysis. These two methods identify genes that are commonly expressed in the condition that we have. Within this commonly expressed gene, it identifies the dispersion of the gene. A volcano plot is a type of scatter plot that shows statistical significance p-value versus magnitude of a change fold value in the volcano plot. All the black dots which we can see in this volcano plot are the genes which are not significantly expressed. Defective doesn't give the list of a background gene. In blue color, those are the genes that are significantly expressed but doesn't show any difference in the fold chain or difference in the expression level. But the expression quantity itself is significantly higher or lower that was noticed. All the red genes are either upregulated or downregulated, depends on the log to fold chain. And we are interested in only red genes. We are not interested in either black genes or either blue genes. The next analysis we do is geo-enriched pathway. Gene ontology enrichment analysis is ubiquitously used for interpreting high-throughput molecular data and generating hypotheses about underlying biological phenomena of experiment. Common pathways between these two comparisons are actin-mediated cell contraction and muscle filament sliding. And there is one more, that is actin-myosin filament sliding. There are quite a number of immune response pathways that are activated, not that are differentially enriched analysis like type 1 interferon pathway. Number of genes related to actin-myosin filament reduced in old adult to young adult. Number of genes related to actin-mediated cell contraction reduces in old adult to young adult, and the gene present in young adult is significantly expressed. Number of genes related to muscle filament sliding are quite the same number in both the cases, that is young adult and the old adult. Further, we can see a lot of ion channels pathways which are suppressed. Cholesterol metabolic pathways which is also suppressed. Regulation of membrane potential pathways are also suppressed. That is very extremely important and related pathways. We conclude from this that membrane potential is not coming back to normal because the balance is disrupted. Regulation of membrane potential is extremely important for the nerve conduction. From this network mapping of GSC analysis, we can see that the transmembrane signaling receptor activity and the response to bondings are identified to be the most important pathways that are differentially negatively enriched in Parkinson's disease versus young adult analysis. There are a lot more pathways or receptor activities which are shown here, but these are the most important and highly negatively expressed pathways. That's why. In case of PD versus OA network mapping, we can say that lymphocytes mediated immunity and adaptive immune response and active matching against pathways are identified to be the most important pathways that are essentially negatively expressed in PD versus young adult studies. So we got differential expressions for both the comparison, that is Parkinson's disease versus old adult and Parkinson's disease versus young adult. For both the comparison, we are recognizing that differential expressed. I mean by that we want this comparison of gene expression to be statistically significant. Second, we want that comparison to make some sort of biological interpretation. For that, we set up threshold for the change, the amount of change that we are observing between the expression of this gene. So, if this threshold is more than one portion, then we mark that gene as upregulated, and if it is less than one portion, then we will mark it as downregulated. And since the biology is a gene, and we want to identify such genes. So here we can see the list of few genes in both the cases which are upregulated and downregulated. Here I used the gene chart to understand the function of the common gene. In case of upregulation, in PD versus young adult, we got 30 genes which are upregulated. In PD versus old adult, we got 139 genes which are upregulated. And in both the cases, we found that the 9 genes are common. And these are the 9 genes which are common in PD versus OA and PD versus Y. I'm talking about left-hand side portion, which the 9 genes include CR, CRYM, CD7, 9A, OPRD1, EPA1A, CD7, APIC1, ECL1, CNTD1. In case of downregulation, in PD versus young adult, we got 41 genes which are downregulated, and in PD versus old adult, we got 88 genes which are downregulated. So in both the cases, that is young adult and old adult, we got 10 common genes which are CSC, NA1E, the second one is METTL21CF, third one is FAM240C, and so on. In upregulation, the genes have functions that we have noticed are mostly related to neurons, muscle contraction, and apoptosis. In downregulation, the genes have functions mostly related to immune response, ion channels, and neural proliferation. Several concepts of interest identified in this study not each have unknown biological roles. The gene signature signaling pattern is very similar between young adult and old adult. Very few of the genes are common. Thus, disease progression and gene risk comparison allow us to make an observation on the trend of the genes that are differentially expressed between these two comparisons and comparing their disease set. We have identified a network of genes that progress with this phenotype and are involved in physiological processes including neurotransmission, wound healing, neural development, cell cycle regulation, ubiquitin exchange, motor neurons, ion channels, metabolic and membrane potential. If it age-matches the sample and thus help us identify and track the difference of expression pattern in these genes to make a proper analysis of the gene data displayed so that we can make further analysis. In the pathway which I mentioned above, we can see many pathways account to be common between these comparisons. They have a good trend, and they have a comparative number of genes that are commonly enriched in the pathways among the top of active pathways. We observe only limited number of pathways that are common. The comparison for a young adult and the old adult so far we've done it for PD versus young adult and the PD versus old adult. By comparing the young adult and the old adult, we can get genes or pathways that are differentially affected or enriched due to the old age themselves, due to the aging process. After identifying such genes, we can eliminate these genes from the whole bunch of genes so that we can get more specialized genes that we have seen in the Parkinson disease. The present data allow us to only speculate or observe the direction of a change. More time, more point, and each lifestyle sample comparison will be necessary to establish proper timing or proper range of observation and observation of a gene by the age. Many of a cancer-related gene could be the result in the old age patient. Further research on the cancer-related gene could be performed. Thank you so much for your time. Thank you for listening to me. Thank you. Goodbye.